Alright folks, how's everybody doing today? I hope you guys are adjusting well, getting ready for spring break. Josh and I have received a number of emails from you regarding lab number 9. So I'd like to take a few moments, address that, the content and the specifics of that lab. So first of all, we've extended the due date on that until March 20th, so it'll be due by midnight on Friday. We've also added the opportunity for you to do a second submission of your materials. So after you view this video, if you decide that you'd like to redo that, you'd like to change your answers or add something to that report, you'd be welcome to do so, and you'd be able to do that quite easily. So many of you have asked in particular how to tell the difference between a magno and parvocellular cell, and I will also cover briefly on-center versus off-center. So the first thing we should do is sort of recreate what you should have for your receptive fields. So we gave you two sets of y-coordinates. And if you plotted y-coordinates number 1 and y-coordinates number 2 for at least one of your cells, it should look something like this. Okay. What we've asked you to do is to take the first set of coordinates and the second set of coordinates and create the difference between them. Each of these curves that you've drawn are technically called Gaussians. And so that green curve is a difference of Gaussians, or a dog, because we've taken the excitatory profile, subtracted from that the inhibitory profile, and what we're left with is this green tuning curve. Okay. Now, if we take a look at this, the first thing we want to figure out is this an on-center or an off-center? So the first question we want to ask is, which portion of that tuning curve, of that difference of Gaussians, is above the x-axis? Anything above the x-axis is going to be excitatory. Anything below that is going to be inhibitory. So it's quite obvious from the outset that the center of that excitatory curve, or of that final tuning curve, is above the x-axis, making it excitatory. If we were to sort of plot downward, we would see that is the sort of size and shape of the center, and we can do something similar with that surround region. And there's the size and shape of your surround. Clearly this is an on-center cell. The center is above that x-axis, making it positive. Let's draw a corresponding off-center cell. So in this case, what you would see is the excitatory region a bit flatter, the inhibitory region sharper. And when you create this difference of Gaussians, you get this similar shape, but it's sort of inverted. In this case, we ask ourselves the same question. We look at that center and we say, is that above or below the x-axis? In this case, we see that it's below. So if we were to draw that, that would be your inhibitory region. And then we can see that surrounding region coming just above the x-axis, which indicates that it's excitatory. So both of these should have similar sizes and shapes, but you see that they're inverted. The cell here is on center. And again, we can determine that by either drawing that receptive field or simply looking at the tuning curve, and we get the same idea down here. We see it's off center, again, inhibitory center, or just look at that difference of Gaussians. And you can see that it goes below the x-axis, and so that makes it inhibitory. Now, what I'd like to do is draw another cell. I'm going to draw another excitatory center cell, so an on-center cell in this particular case. So let's take that excitatory profile. Let's draw that. Let's draw that inhibitory profile. And let's put in our difference of Gaussians. That way we can get an idea about the shape of the tuning curve, or the receptive field shape for this cell. Now, I've drawn these two to scale. Okay, so I want you to see that they have slightly different shapes, at least in one dimension, right? And so if we were to come down with our center portion, and then we were to come down with our surround portion, one thing that we would notice is this is an on-center cell. The center region is above the x-axis, so that makes it excitatory in the center. But we notice that the receptive field is larger than this receptive field, which is smaller. And then if we look at the actual tuning curve or difference of Gaussians there, that receptive field profile, we notice that this one is narrow and this one is wide. And typically when we measure the width of a curve like this, we come down about halfway and select 2 points and measure that distance. And so in this case we're looking at 7 or 8 units, and this is, so maybe we have 3 units here and we have 7.5 units here. So we see that one is wider and one is narrow. Now the real question is, how does tuning curve width relate to receptive field size, and how does that relate to pathway? And so what we see when we look at this is large receptive field. We'll just write it in this color. Or, I'm sorry, a wide tuning curve means there is a large receptive field, and we know that the large receptive fields belong to the magnocellular pathway. If we have a narrow tuning curve, which gives us a small receptive field, we know that's the parvocellular pathway. Now our next question becomes, how does this relate to spatial frequency? Okay. And so what we'll want to do is let's think about different spatial frequencies. I'm going to draw something with a relatively high spatial frequency. And remember, we measure spatial frequency in cycles per degree, right? So we'll assume that this amount of space I've drawn here is 1 degree. And we see we have 1, 2, maybe 3 cycles per degree here at the bottom. And at the top we have 1, 2, 3, 4. Let's say we have, let's erase this last one, and we'll say 9 cycles per degree, for example. Right? And so what we see is this is a high spatial frequency and a low spatial frequency. Now if we think about receptive field size, what we know is that small receptive fields, right, that are corresponding to those narrow tuning curves from the parvocellular pathway are good for high spatial frequencies. And we know that the low spatial frequency which is going to match up best to a larger receptive field, wider tuning curve, are going to be part of that magnocellular pathway. And so now what we've seen is the relationship between tuning curve with receptive field size, pathway, and then how that relates to spatial frequency sensitivity for each of these channels. Now we have both of these channels going at the same time. The parvocellular pathway and the magnocellular pathway are both looking at natural scenes, because natural scenes have excitatory, or have high spatial frequencies and low spatial frequencies. So we need to make sure that we're using both of these channels when we're looking at natural scenes. They cover the whole area. Now, as we go forward, we'll talk about how we use these receptive field sizes and tuning curves and spatial frequencies. We'll talk about how we're going to think about that in terms of shape and object recognition and motion vision as well. We'll see that there's a little bit of a divergence between what the magnocellular and the parvocellular do in terms of motion vision and so forth as we go forward. So hopefully this gets you some good information, so you'll be able to complete Lab 9. You'll be able to think about the tuning curves and those difference of Gaussians that you created, how that size and shape relates to the receptive field structure and which pathway that is coming from. And this is in the lateral geniculate nucleus. All right, well, if you guys have any questions, just let Josh or I know, and we'll be happy to get things straightened out for you. I do hope you enjoy the rest of this week, and also that you enjoy next week's for spring break. Keep in mind we're continuing with our non-face-to-face meetings for several weeks now. Keep checking your email, keep checking Blackboard. We'll communicate with you as we get more information about what's going on. We'll have that sent out to you as quickly as we can. Most of the labs are being prepared and ready for upload on Blackboard, so those things will be available to you in the coming weeks, and we'll have those resources for you as you need them. All right, stay safe and have a good time.